Hi everyone, thanks for checking out this video. So this video is going to be a little bit different than videos I've made in the past. So this is going to be my first video on this topic of bioinformatics sequence analysis. So in my own research in grad school, I don't really do much of this kind of sequence analysis research, but I think it still is important to know how to do if you're working in the bioinformatics field. And it's pretty cool too because in a lot of other fields like experimental biology or physics or chemistry or something, doing research requires a lot of like very expensive equipment and like expensive like physical material. But if you're working on bioinformatics research, a lot of the research you can do actually just involves like free tools that are available online for anyone to use. And today I'm going to be showing you guys how to use one of those free online tools to do some cool sequence analysis research for a protein sequence. Okay, so let's get started. So let's say you get an email from a experimental biologist saying that they found this mystery protein, and they're not sure what this protein is or what it does, but they've just recently discovered this mystery protein, and they want a bioinformatics person to kind of take a look and see if it matches with any known proteins, and basically they just want to see what you can find out about this like mystery protein they've discovered in their lab. And so you get this file from them, this what's called a FASTA file, and basically this file is a sequence of the amino acids in the protein. So each one of these letters here, I know it looks like a bunch of gibberish or something, but each one of these letters just represents one of the amino acids in the protein sequence. So I'll just give you guys a quick review here. So basically, like this is the structure of the DNA. This is just the Wikipedia page here for DNA and RNA codon tables. So this is the structure of the DNA. Remember, it's this sequence of base pairs that can be A, T, G, or C. So the structure of the DNA is just this long sequence of these nucleotide base pairs, and then every triplet of these base pairs is what's called a codon. So these are like the example triplets here, triplets of base pairs. And these triplets correspond to amino acids that are going to be put together when the protein is being translated from the RNA. Yeah, just remember the DNA is transcribed into RNA, messenger RNA, and the RNA then is translated into the sequence of amino acids that makes up a protein. So each one of these codons here, triplets of nucleotides called codons, these correspond to amino acids that are going to be put together to make up the protein. And this table here shows which codons correspond to which amino acids. So for example, and this is RNA, so we have a U instead of a T. If you have some triplet that goes U, C, A, then that would correspond to the serine amino acid. And you can see there's some redundancy. So if it was also like U, C, G, that would also correspond to serine. And then there's some codons that like signal the translation process to stop. But yeah, this is the basics of how a protein gets put together. It's you have some RNA that has been transcribed from the DNA, and the RNA is then looked at in these triplets to correspond to amino acids that are then put together to make up the protein. But yeah, so basically that's all this is here. I know it looks like a lot of gibberish or something, but basically all of these letters correspond to some amino acid here. And this is how you represent a protein in sequence form. So this is what's called a FASTA file. So I'll show you guys this like file format too. It's called FASTA. This is in bioinformatics what's used for representing sequences. It could be a amino acid sequence, which is how you show that's how you represent a protein with its amino acid sequence, or it could be a nucleotide sequence that is that's representing DNA or RNA. So if you see files ending in FASTA, FNA, FFN, all these file extensions, these are the bioinformatics sequence files. Okay, so that's kind of like the basics. That's just an explanation of what this file is that you're getting. You're receiving this from these this experimental biology group, and they want you to figure out what as a bioinformatician you can say about this mystery protein sequence they've found in their lab. So how do we go about investigating this mystery protein? So today I'm going to be showing you guys a really cool free online resource called UniProt. So UniProt is a free online database of protein sequences that is like very easy to get a lot of really good information about really any kind of protein you can think of. So I'm going to be showing you guys how to, first of all, find what this mystery protein sequence actually is, and then how to use this UniProt resource to like find out more about it. But yeah, this is again just like cool free online tool that you guys can use that I think is super useful for people working in the bioinformatics field. So what we're going to use specifically, so you go to uniprot.org, and then from here we're going to go to this thing called BLAST. So BLAST is the basic local alignment search tool, and basically this is just used to search for some sequence you have and align it to known sequences and try to find out what known sequence, if any, it matches to. So basically what we want to do is just copy all this amino acid sequence we have here. Just going to copy it, and we're going to paste it in this box here, and then these are some things you can set to like kind of specific things. This thing E threshold, we're going to see this in the results in a second, but this is basically just like, it's kind of similar to a p-value. It's how many hits you'd expect by chance. I'll tell you guys more about that when we see the results. It'll make more sense when you see the E values for actual matching results. But yeah, you can set like these things, or you can just leave them in the defaults too, which is what I'm going to do. But yeah, you just copy and paste your sequence in here, and then you go to Run BLAST and click that. Now this is going to take a little bit. This alignment is pretty like computationally. It's a big computational task to do, so this is going to take a little bit. So I think I'm just going to probably cut the video here and then get back to you guys when it's finished. Okay, so the search is now finished. Took like, what was it, like 6 minutes, 7 minutes total. So it does take kind of a long time, but like I said, it is like computationally pretty difficult to do. And maybe in the future, I might even make a video about like how the alignment is actually happening behind the scenes, because that's actually pretty cool too. But yeah, so here's the results. So this mystery protein I was showing you guys is the COVID spike protein. So that's this like mystery protein I picked out, something I thought would be like interesting to look and see what we can find out about it. But yeah, so let me just show you guys like what some of these results mean. So we see here we have one hit that was a 100% identity match, and so this is the one that was like, this is what the answer was. I'll show you guys just right here. This is the one that was 100% matched. So it's the the COVID SARS-CoV-2 spike protein. But I'll show you guys there's some like partial matches too. So this one here was like a 76% match. So this one I think is the SARS-CoV-1 spike protein. I think. Let me just check and see. Yeah. So this is the spike protein from the original SARS virus back from like 2003. So this one, it's not 100% match, but it's like a 76% match because they are somewhat related. And yeah, there's some like different entries, entries also for like similar things. But yeah, the 100% match was what I actually picked, which was the SARS-CoV-2 spike protein. And you can see here it shows like what's called the E value. I mentioned this kind of briefly before, but the E value is basically how many hits you would expect with like this level of alignment in this size of a database that would be only due to random chance. So it's kind of similar idea to a p-value, but yeah, the e-value is like, it's basically saying like, okay, with this size of a database, we would expect this many matches of this level of identity due to chance alone. And here it says like 0.0. We know that it can't nothing can really be a zero point 0.0 chance due to chance, but it's so small, it's like such a small decimal that the computer just rounds it to be 0 0.0 for the value. I'm going to see if there's any. Yeah, some of these have like an actual number for the value. Even these are like super low numbers. So basically, all these hits, we can be pretty confident that they're like actual matches that are not really due to chance. Even the ones that are only partial matches, like this one is like a 38.3% match, but we can still be pretty confident that that partial match isn't really due to chance alone because yeah, that's what the value is telling us basically. How many matches like that we would expect to see due to random chance alone with this size of a database. But yeah, so next I'll show you guys how to look at the actual alignment. So to do the actual alignment, like side by side with your mystery sequence and the match, you just over here select your query sequence and then whatever you want to align it with. So select both of those there, and then you can click align. And again, this is going to take a little bit because yeah, these alignments are not really that easy to do computationally. So yeah, it might take a couple of seconds. Okay, so in this case, it's not super interesting because I purposely picked a mystery sequence that would have 100% match. So this is just 100% match. The query sequence matches this SARS-CoV-2 spike protein sequence perfectly 100% because I picked it like that on purpose. But this would be more interesting. I'll show you guys in a second one of the partial matches too. But this would be more interesting really if you had like a real life mystery sequence that wasn't 100% match, but was maybe like a 98% match or something. Because then you can look at where the differences are and what the differences what the differences might be doing. Like let's say for example that the mystery sequence you got was like a new variant of COVID, and the biologist wanted you to figure out like what was the difference between what's the difference between the spike protein in this like new COVID variant compared to like the regular like canonical COVID spike protein sequence. Then maybe you could use this alignment to like pinpoint where the mutation is and what different amino acids this new variant has compared to the old COVID spike protein, and then you could think about like possibly what those different amino acids could be doing in terms of like how effective it is and things like that. You can also look on the side here. You can look at like different annotations. So you can look at like specific motifs. I don't know if any of them will actually come up for this example, but yeah, like glycosylation, yeah, like mutagenesis. You can look at like different parts of the sequence that have been like annotated with different like biological and like biochemical characteristics. So yeah, you can kind of like this is a really good tool for investigating if you do have some sequence that is like not 100% match. Then this is how you would investigate like what the differences are and if they're interesting. So actually, I'll show you guys now. I'll show you guys how the alignment looks if we do it with the partial match. So if we align our query sequence to the partial match, which was the spike protein from the original 2003 SARS virus. So I'll show you guys what that looks like too. So yeah, this actually is more interesting when it's not 100% match. So this one, like I said, it's like a 76% match. And it's more interesting because we can now see like what sequences of amino acids are in the COVID spike protein that weren't in the original 2003 SARS spike protein. So we can see like, for example, this stretch here is part of our query sequence, but not part of the align sequence. So we might wonder like what what if anything is this sequence of amino acids doing here that could be like impacting the infectiveness of COVID, for example. And then we can see too, just some of them, like it's a pretty close match, but it's definitely not 100% match. So we can see like differences in amino acids here, for example, like the stretch here, it's different in our aligned sequence compared to our query sequence. So we might wonder like what these differences mean biologically, and this could be like a starting off point for like further investigating what biological differences might there be between like the original SARS virus and the new COVID virus, for example. Okay, so one last thing I'll show you guys is how much information you can get from the actual like protein entry pages here. So a lot of the proteins that are cataloged on UniProt have a lot of information about them. So we can go to for our matched sequence, we can just look on this page here, and it has a ton of information, like really like. And I mean, this one especially because this is related to COVID, so of course this one is very relevant and has had a lot of information added to it. But even like pretty obscure proteins, you can still find like a lot of good information about them on UniProt. It's like a great resource for learning about like all kinds of different proteins. But yeah, so we can see like there's a lot of information about like what the protein does biologically. So it says like attaches the viron to the cell membrane by interacting with host receptor, initiating the infection. So this is basically telling us like biologically how this spike protein allows COVID to infect the cells by binding to the ACE2 receptor. And yeah, just a ton of like really good biological information like that. And all of it has like citations to like these PubMed entries here are the citations for all this information. So at this point, just going back to our example of being a bioinformatics researcher and getting this like mystery protein in an email from a biologist, this is where we could start actually like collecting some information that we can send in an email back to this biologist and give them some information that we found. We can tell them like, okay, this is the spike protein of COVID. This is what it does biologically. These are all the citations for how we know it does this stuff biologically. Yeah, there's more down here too. There's like the like official names and taxonomy of it. You can see like the taxonomic lineage of this virus. It's a sarbecovirus, which is a type of beta coronavirus, and all these things are like types of some other type of virus. The host is humans. There's a couple other possible hosts, but yeah, the main host is humans. Information about like the subcellular location. Yeah, and then known mutations. This is super interesting. This shows known mutations to the sequence and what they can do biologically. And again, each one has you have your reference here for how we know about it. But yeah, this is showing like sorry, this is showing like for the sequence all the possible known mutations. And this is saying like at position 121, for example, the amino acid N could be mutated to the amino acid Q. I forget what these stand for, but again, these letters just stand for some amino acids. And we can even see it like graphically where it's occurring in the sequence, and then what the mutation does, and then the citation for it. So yeah, again, this is like super useful if you just have some mystery sequence that you want to do some research on, and then get back to this biologist about what you find. This is like great resource. And then so one more thing I'm going to show you guys is how to actually download the protein sequence. So to download it, we're going to go to format. And you can download it in a couple different formats, but like I said, like FASTA is this like widely used format in bioinformatics for sequence data. So we go to format, FASTA, and then yeah, this is how we get the actual sequence. So yeah, this is how I came up with the original sequence that I like showed you guys in the beginning of this video. I just did that, and then you get this like line of information in the beginning, but I wanted to keep you guys in suspense, so I actually like took out this line of information and just put mystery sequence up here. But this is how you get the actual like FASTA format uh, amino acid sequence. You just go to format and then FASTA. And then if you want to download it, you can also like download. I mean, in this case, it's kind of easy because it's not really that long of a file. So you could even like just copy and paste this into some like text file or something. But let's say you have like a really long FASTA file that you want to actually like download. To do that, you're going to go to add to basket, and then you have your basket here, and then you're going to select which one in your basket you want to download. And then you go to download. And then you can, if you're downloading a lot of them, you might want to compress the file, but here I'll just leave it uncompressed. Then you can again like select the format. I'll just go with FASTA for now, but you can choose a couple different formats. And then you click go. And then yeah, then you get this basically the same thing I showed you in the browser. You just get like a downloaded version of that. And yeah, this is how I got this original mystery sequence here. I just took this downloaded version and then just took out this information line and kind of put mystery sequence up here. But yeah, so that's how you'd get like the actual sequence of a protein from the UniProt entry here. Just remember, go to either like format FASTA or add it to your basket and then download it from your basket like that. But yes, that's all I have for you guys today. So thanks for watching, and if anyone has any questions, just let me know in the comments and I'll try to answer them. But yeah, thanks for watching, and see you next time.